Okay, let us start. What should we do today? Today, shall we start the new chapter, or do you have any doubt in the previous chapter?
Ma'am, please do.
What happened, Priyadarshana? What were you saying?
Ma'am, please revision.
I can't hear you. Can you type on the chat box? I can only hear something. I can't hear properly.
Ma'am, please do revision.
Okay, revision. Okay, yeah. I was asking you that if you want revision, I will do the revision today because your exams are near. So we will just go with the revision. We will not do the next chapter. So which chapter you want first? Like from starting itself, should we revise, or if you have one particular chapter which is tough for you, then we can revise that first. Then we will go with the other chapters. Anything like that, or should we start with the first itself?
Ma'am,
Why
from starting.
Okay. From starting we will revise. Okay, I don't think you need a revision of cell. Do you need? It was in your ninth standard. Shall I?
From second chapter,
Yeah. Okay.
revision start.
Okay. From second chapter we will revise. Chapter number 2, structure of chromosomes, cell cycle and cell division. Just a minute. Take out the page number 8. Page number 8. Okay, we will revise. So, chapter 2, structure of chromosome, cell cycle and cell division. We are doing the revision class. So, pay attention. If you have any doubt, that will be clear from this revision session. So, you all know all body cells are the result of mitotic division. Now, what is a mitotic division? Can anybody answer this?
Ma'am, what?
Mitotic division, or we can say mitosis. We have two types of division, mitosis and meiosis. So what is mitosis? Do anyone remember that? If yes, they can define mitosis. If no, then I will define it. Okay, so I will define what mitosis is. So when you study structure of chromosome and cell division, you should know what is mitosis, because mitosis and meiosis are the two processes taking place in cell division. So you should firstly know what is mitosis. So when we define mitosis, how can we define mitosis? It is the division which results in the production of two daughter cells, which will have same number of chromosomes. That means same number and kind of chromosomes. What is the definition? Division that results in the production of two daughter chromosomes or two daughter cells, each having same number and kind of chromosomes. Okay, is this much clear?
Yes, ma'am.
Yes. So what do you understand? This is very simple. From the definition, it is clear that this division leads to the production of two daughter cells. Okay. Whereas in meiosis, we have four daughter cells. Here in mitosis, diploid cells are produced. In meiosis, haploid cells are produced. Haploid means more than two. They are four in number. But here in mitosis, when we talk about mitosis, still we are talking about mitosis. Mitosis is the result of two daughter cells. It gives the result of two daughter cells, and each daughter cell will have same number and kind of chromosome. That means chromosome number and kind will not be changed there. That means what is the term given when the chromosome number is not changed? Chromosome is similar kind and the number is not changed. Homo. We use the term homo, homozygous. Okay. So the chromosome here is of same kind and number. And the cell divides into two genetically identical daughters. In mitosis, cell which is dividing gives us two genetically identical. That means they are identical daughters. Why? Because the chromosome number and the kind is similar. They are not changed. Homozygosity is there. That's why they gives rise to two genetically identical daughter cells. So this is mitosis. So as we all know, all the body cells, the cells which are present inside our body, is the result of what? Repeated mitotic division. Okay, we have studied the process of mitosis and meiosis. Mitosis is a repeated division. It has some phases: anaphase, prophase, telophase, etc. So after covering all the phases, it again starts with the first. So it is a repeated division, right? Now, what is the difference between mitosis and meiosis? I told you mitosis gives two nuclei, or we can say two cells. That means we get two genetically identical daughter cells, which are two in number. Just remember this number. Mitosis gives us two, whereas meiosis gives us four. Then mitotic gives identical cells to each other and to mother cell, while meiosis leads to genetic variation. Okay, mitosis, I already told that mitosis gives us genetically identical individual, whereas meiosis, there are chances of genetic variations. Why there are chances of genetic variations in meiosis? Because in meiosis, the process of meiosis, crossing over takes place. Crossing over and independent assortment takes place. So that's why meiosis have some genetic variations, whereas mitosis leads to genetically identical cells. Okay. Then talking about crossover, what is crossing over? We have discussed that meiosis leads to genetic variation because of crossing over. Now, what is crossing over? Crossing over is nothing but exchange of genes. Okay, exchange of genes in that particular chromosome. Which chromosome? A homologous chromosome, same structured, same kinded chromosome, and it will result in a mix of parental characters. Okay, not a single parental character, but it will result into a mix of parental characters which will be present in the offspring formed. Is it clear? Crossing over?
Yes, ma'am.
Yes. Okay. So what is okay? I think crossing over is there in your book, or if it is not given, you can just write the definition of crossing over. I'm not reading the whole chapter. There are many long paragraphs in this chapter. I'm not reading the chapter. I'm just revising the main topics which will come in exam, which are important to know. So you can write what crossing over is. Just a minute. I'll share my screen and I'll.
Ma'am, it is written crossing over definition.
Okay, it's there in the book. If it's there,
Ma'am,
then
page number 17.
page number.
17.
Okay, page number 17, crossing over is there. Okay, fine. Just a minute. Yeah, it's there. Okay, page number 17, right hand side, significance of meiosis. Second point, second subpoint. While the maternal and paternal chromosomes are separating, the chromatid material very often gets exchanged between the two members of homologous pair. This is known as crossing over. So, same thing, only my definition, words were changed, but it's the same thing. Okay, crossing over is the exchange of genes between the homologous chromosome, which will result in the mix of parental characters in the offspring. That is crossing over. Then what is independent assortment? Independent assortment is the process which defines how genes independently separate from each other. Okay, genes separate from each other during these processes. So the independent separation of genes is discussed in independent assortment. Okay, in independent assortment, the independent separation of genes from one another is discussed. Then we're talking about mitosis. It gives four haploid, sorry, two, mitosis gives two and meiosis gives four. Yes, don't get confused. Mitosis gives nuclei with same number of chromosomes as mother cell, while meiosis gives half the number. We are discussing the difference between mitosis and meiosis. Okay. So first, we told that mitosis have two nuclei or two cells, and meiosis gives rise to four cells. Then mitosis gives identical cells which are identical to each other, and meiosis leads to genetic variation. That means due to crossing over and independent assortment, it leads to genetic variations. Genes get exchanged. That is known as crossing over. Then we are talking about mitosis. It gives nuclei with same number of chromosome, whereas meiosis gives half the number. Okay, meiosis results to 4 cells. So all the 4 cells are getting half the number of chromosome. Okay, whereas in mitosis it is just 2, so it will get equal number of chromosomes. Then fourth point, mitosis includes yes, whose mic is on? Yes. So mitosis includes one division and meiosis includes two. There are two divisions in meiosis. What were they? Can you remember? Meiosis is divided into two. They are known as karyokinesis and cytokinesis. That means nucleus and the cytoplasm, both of them divides. Okay. But here in mitosis, only one division is there. One division is there in mitosis. Now, okay, just a minute. I can just show you with the help of a flow chart if you want. I have already shown you with the help of flow chart, but I'll repeat this so that the process or the method will be more clear to you. Just a minute. Yes. So mitosis. Mitosis is also known as the M phase, mitotic phase. So suppose this is a cell. This is the nuclei inside. Now mitosis gives out how many cells? Two identical daughter cells. Like firstly in mitosis, you can see this is the nucleus and cytoplasm divides. It will form chromosome. It will form different chromatin fibers can be seen in the cell. Okay, different chromatin fibers can be seen in the cell like this. Then it will give rise to two daughter cells, two identical daughter cells. Okay, this is the two identical daughter cell. This is the nucleus inside. So this process is known as mitosis. This is mitosis. Yes. And then mitosis or the mitotic phase, M phase. Then talking about meiosis, the meiosis is divided into two phases. Basically, mitosis and meiosis both have the division of this one, nucleus and the cytoplasm, but meiosis divides in two ways. That means we have meiosis 1 and meiosis 2. So this is meiosis 1 where the cell, okay, we can see some chromatin fibers inside. Again, like mitosis, one more cell is there with chromatin fibers inside. Then it will first give rise to how many cells? Two cells. First, it will give rise to two cells in meiosis 1. Okay, parent cell divide twice. Okay, this is the parent cell. Two cells, they are the parent cell. Now this will again give rise to two more. So total how many? 1, 2, 3, 4. Total 4 cells are formed. So this parent cell, which are the parent cell here? This one. These two are the parent cell. This will divide into two each, and at last we will get 4 cells. That means parent cell is dividing twice. So from this parent cell, half is going here, half is going here. So that's why we are saying in meiosis, only half of the number of genetic material or half of the amount is transferred. It's not like mitosis. Mitosis, they get full amount of genetic material. Okay, that's why they are identical to each other, genetically identical. But meiosis, it is meiosis 2, M2. So that's why when meiosis 2 happens or when it occurs, it divides into 4. Then one parent is, giving rise to two cell. Half genetic material is here and half of them is here. So only the half material is transferred to the offspring or the divided cells. Is it clear?
Yes, ma'am.
Okay. when you try to learn for exam, just try to make these flow charts and learn, so it will be easy for you. When you go through the paragraphs, it will all be a mess. You will not understand anything. So if you make flow charts and learn, it will be easier for you. So now you all know this basic thing, that first we have a single cell. Okay, like in all the mitosis, meiosis, one single cell is there. It will divide into, okay, sorry, it will have a nucleus each. Whether it's a one cell, two cell, every cell will have a nucleus. A nucleus contains of what? Chromosomes. As we have seen the structure of a nucleus. This nucleus contains chromosomes inside it, like this. Okay, so nucleus will contain chromosomes. Then chromosome will contain last DNA. You all know the structure of chromosome is also discussed in this chapter. So chromosomes contain DNA. These chromosomes have DNA threads in them. So they are known as chromatin fibers. So the chromosome is, chromosome takes place or takes rise with the help of DNA. So this is the sequence. Cell contains nucleus, nucleus have chromosomes, chromosomes have DNA. This is the whole sequence. Okay. You can make the diagram also. This is the cell. Then innermost, it has the nucleus. This is the nucleus. Then inside it, some thread-like structure. This is the chromosome, and chromosome contains DNA. Is it clear?
Yes, ma'am.
Yes. So you can make the structure for your better reference. Then we all know in human, how many number of chromosomes are there? Total 46 chromosomes are there. Right. Have you all made this structure? Are you making this, or shall I rub this? If you want to make this, you can make cell, nucleus, chromosome, DNA. Then you can make this structure. The outermost is the cell. Okay. Inside it, it has a nucleus. Nucleus contains chromosome. Chromosome is made of DNA. Clear? I think it's clear to you. Okay, I'm rubbing.
Yes, ma'am.
Yeah. Yes. So in human, you all know 46 chromosomes are there. Okay. And how many pairs are there? 23 pairs. 23 pairs are there, and total 46. So now 23 plus 23 is 46. So you all know, we have already discussed the pair 1 to 22 is known as autosome. Now autosomes, they are basically present. What is their function? What is the function of autosomes? They just do the different functions in the body. Okay, apart from the sexual purpose, other functions are done by autosomes. Autosomes. Then this is pair 1 to 22 is autosome. And the last pair, which is the 23rd pair, is known as sex chromosome. So this 23rd pair, which is the sex chromosome, is responsible for the sexual characters, or the change in the sexual characters, the genetic identities. This pair, this 23rd pair, is responsible for the sexual matters inside the body. Okay, that means this 23rd pair determines the sex of the child. The child which is formed, the offspring which is formed, the sex is determined by the help of this 23rd pair of chromosome, which is the sex chromosome. All the other 1 to 22 are autosomes. They do not have nothing. They have nothing to do with the sexual processes. Okay, so you all know, for boy we use XY chromosome. For girl we use XX. So this 23rd pair, if this 23rd pair is XY, the child will be a boy. Okay. And if it is XX, it will be a girl. Clear? Is this clear?
Yes, ma'am.
Yes.
Yes,
So,
ma'am.
yeah, your book have a chart, I guess. It's not here. Just a minute. Okay, we will discuss it later. I think there is a chart in the next chapter, or I don't know where, in genetics, I think. There is a chart of XX chromosome, XY chromosome. So there, this 23rd pair is determining the sex. If it is XY, it will be a boy. If it is XX, it will be a girl. Then, moving forward, yes. Now, you all know chromosome inside a nucleus, chromosome is present. Now, these chromosome contains chromatin threads. Now, you know during division, these chromatin threads, they condense. Okay, they condense and they look like this. Okay, they look like cross structures, chromosomes. Okay, due to condensation of chromatin threads, they form this X-shaped structure which is known as chromosome. These chromatin threads, these are the chromatin threads during mitosis during division, or we can say during cell division, these chromatin threads condense and they form these X-like structure. These structures are known as chromosomes. These are the chromosomes. Okay. Now we all know during this cell division they form exact copies. That means exact replica. Replica means copies. So during cell division, these chromatin threads forms exact copies that are known as chromosomes. They look very similar. They are exact copies of each other, or we can say exact replica of each other. So they are, at the end they are known as chromosomes. Then these are some basic things. You should know this, that you will not get confused during exam when a big question is asked, like define mitosis and show the process, all the processes involving in mitosis. You will not get confused if you know that I already told you chromatin threads condense during cell division and gives exact copies which are known as chromosomes. So this you can also write when you write all the phases of mitosis. Then okay, we already told these chromatin fibers during cell division, they are known as chromosomes. They form these X-like structures known as chromosomes. Now these chromosomes are individually known as chromatids. Like this one is different, this one is different, they are individuals. So these are individually known as chromatids. Then that means one individual strand. You can say this is one strand of chromosome. Here is the centromere, the central portion. And this is the second strand. So this one single strand is known as chromatid. This is also a single individual. This is known as chromatid. Okay. And when they form together, that means this and this two. Okay. When they fuse together and they form this one, this is known as a chromosome. Okay. Some basic things I'm just repeating. I hope you are getting my point.
Yes.
Yes. So this single structure, these single line are known as chromatid. When they fuse, when they form a X-like structure with a centromere in between, this is known as chromosomes. Okay. Then we have the cell cycle. Just now we discussed about structure of chromosome. Now we have the cell cycle. So we already told structure of chromosome contain DNA. Along with DNA, one more thing is present. What are they? Histones. Histones are nothing. They are proteins present along with the DNA. Okay, you have to remember this thing. Histones are the proteins which are present along with DNA. Now we are not going so deep. You all know a DNA structure. How does it look like? We have a DNA strand like this. Okay, and we have bonds between them. That means we have four types of, DNA consists of what all? Single DNA strand is composed of nucleotides. So we have all the nucleotides and sugar and phosphate and the nitrogenous bases. So we have The two strands together make a ladder. Okay, four types of nitrogenous bases. There are four types of nitrogenous bases which are found in the DNA strand. Okay, I'm not writing. So what are the four types? Adenine, cytosine, thymine, and guanine. These are the four nitrogenous bases. Along with these nitrogenous bases, nucleotides and phosphates are also present. Phosphates, sugar molecules, etc. all are present. Now you all know adenine bonds with thymine. With how many bonds? Double bond. And cytosine bonds with guanine with a triple bond. Okay. Yes?
Triple bond.
Yes, triple bond. The cytosine bonds with guanine with triple bond and adenine bond with thymine with double bond. Okay. Then talking about the structure of the DNA, the first is guanine cytosine bond, C G with triple bond like this, C triple bond G. Then second the space we have adenine thymine. Okay, A double bond T. Then again followed by CG. Sorry, again we have two adenine and thymine. That means again T A like this. And then we have G C. Okay, that this is the bonding. Okay, between one CG pair, we have two adenine thymine pair like this. G C. Between them, we have two adenine and thymine pair. Okay, it can be AT or TA. Clear?
Yes, ma'am.
Yes. So just go through the structure of this DNA once. Later, you can go through this. So this we have talked about DNA, structure of chromosome we have discussed. Okay, when they condense, they form the chromosomes and the single strands are known as chromatids. This is the structure of chromosome. Now we have the cell division. The cell cycle. So cell cycle has a saying that means divide, grow, and redivide. In the cell cycle, the cell first divides, then it grows, and then it redivides. So this is the saying of cell cycle: divide, grow, and redivide. Okay. Now, cell cycle has some important task in it. Okay, the cell cycle, divide, grow, and redivide, consists of some important tasks. What are these tasks? Growth, then protein synthesis and synthesis of RNA, then duplicating of DNA before splitting into daughter cells. Okay, like you all know, one parent cell splits into two daughter cells. So the duplication of DNA is very necessary here. Then only one will go here and one will go here. Otherwise, only either of them will get the DNA, not both of them will get. So the duplication is required in the first stage when it is in the parent stage. Duplication of DNA is required, then the DNA, half of the DNA will go here, the first daughter cell, then the half will go to the second daughter cell. Other things I already told you, growth, synthesis of proteins and RNA is required in during the cell cycle. Clear?
Yes, ma'am.
Yes. Then we have, now we will discuss the cell cycle in a brief way. So you all know cell cycle is divided into interphase and mitotic phase. We have two phases, the interphase and the M phase, mitotic phase. So interphase, if you remember, interphase is the non-dividing phase. It is non-dividing. And the mitotic phase is the dividing phase. DP stands for dividing phase. Okay. So non-dividing means you cannot say interphase. In interphase, nothing is going on. In interphase, since interphase is a resting phase, okay, it is non-dividing phase, so we can say it as a resting phase, but we cannot say it is totally a resting phase. It is quite active. Why? Because in interphase, some protein synthesis and synthesis of RNA, DNA, everything is taking place. Okay, it is quite active because synthesis of DNA is taking place. Basically, we require DNA. Okay, DNA is required in the chromosome. So apart from RNA and proteins, main synthesis of DNA is done in this interphase, DNA synthesis. Okay, so we cannot say that it is a totally resting phase. It is a resting phase, but quite quite active processes are going inside, which is the synthesis of DNA. Clear? Okay, I hope this is
Yes,
clear.
ma'am.
Yeah. So interphase, we all say interphase as a resting phase, but it is not totally a resting phase because DNA synthesis is taking place in that resting phase. Then, now talking about interphase, during this phase, what happens? The cell just prepare themselves for the next cell division. Okay, cell is preparing themselves. How they are preparing? We already know, by synthesizing the DNA. Synthesis of DNA is taking place and they are preparing themselves for the next cell division. But there is no change in the chromosome. That means chromosome number, kind and everything, the structure will not be changed. Only they are synthesizing the DNA inside the chromosome. Clear? Don't get confused. The chromosome is not changing. Only the DNA inside is getting synthesized. Then we have, okay, again when we talk about interphase, we have three phases. First growth phase, then synthesis phase, and then second growth phase. GP is the growth phase, this SP is the synthesis phase, and this is the first growth phase. First GP is the first growth phase, synthesis phase, and the second growth phase. So these are the phases of interphase. Now interphase is further divided into three phases: first growth, synthesis, and second growth phase. Now what is happening in the first growth phase? If you remember, RNA and proteins are synthesized here. Okay, RNA material and the proteins are synthesized in this first growth phase. So in this phase, what happens? Either the cells will go into resting phase or they will start preparing for the next cell division by synthesis phase. That means some cells, it depends. In the first growth phase, either the cells will go into resting stage and some will go into synthesis phase for further synthesis. Okay, now here in synthesis phase, what is synthesized? More DNA is synthesized. In the first growth phase, what was synthesized? RNA and proteins were synthesized. From these RNA and protein cells, some cells will go into resting stage and some will go into synthesis phase. Now in this synthesis phase, more DNA is synthesized and chromosomes get duplicated. That means chromosomes, they are duplicated and form more in number. Okay, now these chromosomes gets duplicated in the synthesis phase and they form more in number. Now in the second growth phase, RNA and proteins which are necessary for cell division are again synthesized. Okay, first growth phase also RNA and proteins were synthesizing. Second, in the synthesis phase, DNA was synthesized. And the second growth phase, again the RNA and proteins which are necessary for the cell division, they are getting synthesized. RNA and proteins are getting synthesized. Now, this is the interphase. Interphase divided into three phases. I'm repeating. First growth, second is the synthesis phase, then second growth phase. In the first growth phase, what is happening? RNA and proteins are synthesized, and some cells go into resting phase, and the others go into either. There are chances. Some will go into resting phase, some will go into synthesis phase. Now, the ones which have entered the synthesis phase, the DNA synthesis takes place. There, more DNA is synthesized, and the chromosome will get duplicated. These are the duplicated chromosomes. Then from here, they will enter the second growth phase, where again the cells are required for the cell division. RNA and proteins are synthesized there again. Okay, is this much clear to you all? Shall I repeat anything? Everyone, is this much clear? Astha, Abhishek, are you following me?
Yes, ma'am.
Yes. So is this much clear to you all? I'm asking you.
Yes, ma'am.
Yes, if you have any doubt, please stop and ask me. I'm rubbing this. So this we talked about interface. Okay, I think we are doing the revision. That's why all are left. All have left the class. So yeah, time is also over. We have 3 minutes more. So we will discuss the next thing in the next class. Okay, that means after interphase, this cell or these cells are entering the dividing phase. Now, what is the dividing phase? The mitotic phase. After interphase, the cell is reaching to the M phase, which is the dividing phase. Now, interphase, we already discussed 3 phases. After all the protein synthesis and the DNA synthesis, now these cells are entering to the M phase, mitotic phase. Now here what happens? New cells are formed. New cells are formed here in the mitotic phase. And from here they are again entering the interphase here. Okay, and the cell cycle continues. This is very easy. Okay, from mitotic phase, new cells are formed. These newly cells formed again enters the interphase, and then this cycle continues. Again interphase, growth phase 1, synthesis phase, growth phase 2, then again mitotic phase, then again new cells will be formed. Again they will go into interphase, and this process continues. Is it clear? I hope this is clear to you all. Okay, so we are stopping here. We will do the next portion in the next class. Clear?